Today's lesson is about interpretation of results, which is the fourth and last layer, last level of our subdivision of the research problem. Let's say that we have run the experiments, we have generated data, we have thus results, but we need to interpret them, because results are not always straightforward in their interpretation. They do not always conform to expectations, and so it is necessary to analyze them, sometimes in depth. Sometimes it takes more time to interpret the results than to generate them. I'll show you a few examples. So, to start, we can say that an experiment generates answers in the form of qualitative or quantitative data. Qualitative data occurs in the form of yes or no, black, red, white or green, etc. Quantitative data are always expressed in numbers, often measurement units, but anyway, they express quantities. Whenever possible, we prefer to have quantitative data. It is not always possible. So, I'll always forget this. It takes a second. So to reinforce what we have just said, we'll say now that data do not speak for themselves. They must be analyzed. What are data? We tend to think of data as numbers. Not necessarily. Especially in English, data refers to whatever is generated as the results of observations or experiments. So if you have, say, microscopic pictures, those are data. If you have spectra, those are data. If you have, I don't know, photographic films, those are data, and so on. So I'd like to show you again this little analysis that we did some time ago, and that I already showed you in a previous lesson, and that on statistics, in which, if you remember, at first sight, these columns suggested that the data, that the results were quite different in the different conditions considered. But then, on looking a bit harder at the graph, one realizes that the differences between the columns are really very, very small, in the order of thousands of units. So, very minor differences that are simply enhanced by the particular tool used to graph them. And so it is necessary to think carefully about the data in order to interpret them properly. If we have just looked at this graph, draw a conclusion that there are differences, this is certainly the condition in which we have the smaller signal, this is the higher signal, that would have been wrong. But this is, after all, easy. There are instances in which the data really require hard thinking, such as this one. This is a very small fraction of quite old experiments run in my laboratory, when it was about 2004 or so, at the beginning of the microarrays era. That is when next generation sequencing was really not available yet, and the way to address global gene expression, that is expression of all the genes in a given biological sample, whether cells, or tissues, or organs, whatever, was using microarrays. Microarrays came in a variety of forms. Basically, you hybridize the RNA extracted from your sample to an array, which is a physical object in which oligonucleotides recognize the different DNAs, and depending on the intensity of the fluorescence or of the signal emitted, you can determine how much a given gene is expressed. The more the RNA, presumably, the more the gene is expressed. Okay, so in this particular chip we had some 20,000 genes, many of which were represented by 2, 3, or more oligonucleotides. So we had quite a few tens of thousands of rows in the sheet, so this is a very small fraction. Each row was longer than this, contained many data, and basically what we were doing was to compare two treatments: a control treatment, that is a negative control, and a specific treatment, doesn't matter what it was, in a given cell line. So we did the experiment the first time. It was the very first time we did such an experiment, and there was little experience all over the world because the microarrays were new. And as guidance, well, you had different parameters here. Basically, here you had the signal of each specific gene listed here and described here on an arbitrary scale. Then you have a statistical evaluation that says whether the gene is absent, A, or P, present, which means it is expressed or not expressed, or rather detectable or not detectable, depending on the sensitivity threshold of the method. And you can compare two situations, or more, in this case only two. The situation called CRE, which was our test, and the situation called ECO, which was our negative control. Okay, so we compared all genes in the first experiment, and we found that several of them, many of them, were upregulated by this condition here, and others were downregulated. So that was interesting. But before analyzing the meaning of these genes, what these genes were telling us, we decided we wanted to repeat the experiment at least 2 other times in order to determine whether it was reliable, or that means it was repeatable from one time to the next, and whether we could really trust the results, hence. Because, of course, if the experiment shifts every time you run it, you cannot really trust the results, and the interpretation becomes very volatile. So we did the experiment 3 times, and here is the synthesis of 3 experiments. Now, this is, again, a small fraction, but a smaller magnification of the Excel workbook that I've shown you before. And here we have columns representing the 3 experiments. Each row, now unreadable, is a single gene. These are the 3 experiments, vertically aligned. Red means increased, green means decreased, white is unchanged. So you see that in the 3 experiments there were genes whose expression was increased in 2 experiments but unchanged between the 2 conditions that were compared in the 3rd experiment. At times it was up in one experiment, down in another, unchanged in another. Here, up, unchanged, up, unchanged, down, etc. there is very little repeatability. This was really a very annoying result, because we thought we must have done something really wrong. We don't understand it, and we don't have the possibility to find out, because these experiments were rather expensive, well, very expensive at the time, and we couldn't run tens of them. So that was really upsetting. But before throwing everything away, of course we analyzed the data very, very carefully. And it took really days of very serious thinking and analysis, especially because the tools at the time were represented by an Excel worksheet, which you had to rearrange the data, and because there were so many and the computers were not so powerful, it took a few minutes every time you were doing a transformation. So you had to really think about what you were doing, then set up the necessary formulas, make sure you didn't mess up, because if you mix data on an Excel worksheet, there is no coming back, you are lost. And so you have to start it all over again, assuming you have kept a copy of the originals. If you haven't, you are lost, and that's it. So it was really hard work, and initially we couldn't understand. Here was the situation in which we compared two experiments. Here, condition A was compared with condition 2 in experiment, with condition B in experiment 2, and here condition A was compared with the same condition B in experiment 1. So these are two experiments. And you see, there is no correlation whatsoever. If you take into consideration the ratios of each gene, each dot here is a gene. So you say, you ask, what is the ratio of gene X in condition A versus gene X in condition B in experiment 2 and in experiment 1? And you find that it is random. Sometimes they agree, sometimes they don't, sometimes there is no regularity here. In fact, the dots here are spread all over in an apparently symmetrical fashion. If they were highly correlated, they would run like this. It would be on a line.
R2 squared is really, really small, like 2.
Yeah, it's basically 0. Exactly. And in fact, the linear regression line is horizontal. Okay, but there was something that spoke in a different way. If we correlated condition A in experiment 1 with condition A in experiment 2, there was an excellent correlation. So the experiments were repeatable. If I had a high number, a high result for one gene in experiment A, it was also high in experiment 1, it was also high in experiment 2. If I had it in 1, it was also in 3, and so on. These are all the possible comparisons between the three experiments. And if you do the same with B, with condition B, it's the same. There's a very high correlation. So, how come the results themselves are very repeatable, and yet the comparison, the ratios in the of the expression of each gene in the two conditions vary so much in the different experiments? It took quite a while, and I want to go through the whole analysis because it would be long. It would take your time away. I just want to come to the conclusion, which finally we found when we rearranged the data in this way. Here, this is a particular experiment, number 1, but it's the same with all three experiments. This is a ratio between condition A and condition B for each gene. So again, each dot is a different gene. And this is represented as a logarithm base 2 of the ratio. So 2 means 4. 4 means 16, etc. And this is instead 1 half, 1 fourth, 1 eighth, etc. And now you see the shape that the dots take. They spread quite a lot down here. But the relative minority of them are aligned on the 0 line. And 0, 2 to the 0 means 1. So these dots here have a ratio of 1. These genes are not changing expression. What apparently changes expression are the genes that have low absolute intensity. The genes that produce low numbers on the chip, on the array. So, the interpretation, I'm asking you the interpretation. It is not easy, but try to understand what this means. So, I'll explain again what you think.
Yeah.
If you have low intensity of expression, the dots spread all over. Sometimes are very increased, or the ratio is very high. Sometimes the ratio is very low, and there is apparently no relationship, no regularity. They different genes spread in a different way in different experiments. But these genes here stay here in the middle in all experiments. So what does this mean?
That my tool is no, so it cannot detect under
A certain
yeah.
threshold.
That's right.
Yes. So if you put, say, an arbitrary threshold here, and you only consider the results from here up, the results are very consistent. There is very little gene expression change in the two conditions, A versus B. What apparently changes is low intensity genes whose apparent intensity is close to the background. So they basically oscillate in a way that is unpredictable because it is highly affected by the background and or by the sensitivity of the tool, of the instrument. What this also means is that the sensitivity of the arrays was too low to allow us to analyze most of the genes. Only relatively few were analyzable. And those didn't change between the two conditions. So, this is an example where initially you have a situation that makes no sense at all. And then, while after analyzing it carefully and trying to understand what the data are telling you, you reach finally a conclusion. Maybe not the conclusion we are hoping for, but at least you understand what the data are saying. So, analyzing the data is always important. I've been many times in situations when a younger person in the lab came in and said, this experiment is crap, it's not working, you see that the data make no sense. And then I sat with him or with her and analyzed the data, and I go, they mean something. It's just a matter of interrogating them properly. So, data do not speak for themselves, they must be analyzed, we have said that. And the answers we get are not always welcome, like in the case I've just shown. But a valid experiment always tells us something. So, this is a lesson that you should always remember. Do not discard an experiment unless you know it is faulty, unless you know you have made a mistake. There are times when it is very obvious that the experiment went wrong. But otherwise, you should not discard data. You should strive to interpret them. If you, with all your efforts, cannot interpret them, set them aside, and think about them later, maybe even years later, but do not discard them unless you know that the experiment is on. Because a valid experiment always tells something. And drawing from a previous lesson, one of the first we did, you know, when we are looking for something, we tend to disregard things that do not align with our conviction. So at times we tend to consider wrong, bad data that instead are telling us a truth we don't want to hear. So be very careful. Except that there is a very long tradition of suppressing data that scientists don't like. This is Pythagoras, who allegedly, according to the legend, killed the poor guy that discovered the irrational numbers. Because Pythagoras was a mystic who thought the universe was made of numbers. He could not stand the existence of irrational numbers. And so he didn't want the news to be made public and kill the guy who they discovered. But this is, of course, not the proper way in which scientists should behave. Most of the times we don't kill people anymore. All right, so again, on the interpretation of the results, we haven't really defined what a result is. I'll try to explain what it is using this little schematic. The data are the raw output of your observation of your experiment. In the case of the microvasils that I've shown you, the data were the numbers that I was reading on the Excel workbook. The results are rather the immediate conclusions that you can draw from the data. Oftentimes, it is probably easiest to define results in this way. Oftentimes, the data are generated to validate a hypothesis. So, the results are the logical consequence of the data. Once you have analyzed the data, the results are basically, is the hypothesis validated or not? Those are the results. And those are so immediate and so mostly based on logics that that they don't need much of an interpretation. Results stem directly from the data, provided that you have analyzed the data. Conclusions, instead, are quite more complex. Conclusions relate to the need to generalize your results. Your results might be, certainly, we'll talk about the particular plants that you're using in your greenhouse, or the particular cells that you're growing in your laboratory, or the particular animals that you use for experiments, but they won't tell anything directly about different plants, different cells, different animals, different treatments, different laboratories, different scientists, etc., etc., etc. But of course, if you were to stick to the results, you would have to draw very, very extremely narrow conclusions, such as A causes B in this cell line, in my laboratory, in my conditions, on Thursday, because I've done the experiments on Thursday. But nobody would care about conclusions like this. So you are forced to generalize. So first of all, you would conclude that probably the results would be the same in any day of the week. That would be one thing. But then you would try to make more risky generalizations, which are nonetheless necessary. You will try to say that, well, if my cells were human, probably my results apply to all human cells, or at least to all human cells of this kind, not necessarily, not only to my own little cell. Or we might even say, Well, they apply to all mammals, or to apply to all animals, or to the whole tree of life, whatever. And the question is, first of all, who allows you to generalize? What authority you have to generalize? Until you stick to your data and to your results, you are okay. You have strong support for what you're saying. But then, how do you generalize? You've said that you need to, you have to, because people are not interested in extremely narrow results. And yet, once you generalize, you are no longer backed up by data, by hard data at least. So, there are two main pillars according to which you generalize, and that's a matter of judgment. You have to try to walk on a very narrow pathway. That is, you should generalize as far as possible, but not beyond that point. And you don't really know where the point is. So basically you rely on knowledge of the field, whatever is previously known, whatever generalizations have previously worked, have been previously supported by subsequent data. So you know that generally that kind of generalization is allowable, makes sense. And second, you are supported by your experience. Experience, of course, is something that you have to build up. Once you're young, you have very little experience, and then later on you accumulate experience, and you will have been before in similar situation. You will have generalized in a similar way before, and you know that it's fairly safe to generalize it that way, and not beyond a certain point. So, whenever we draw conclusions, though, there is a strong degree of judgment on our side. And this is one of the reasons why conclusions are never unique. Different scientists analyzing the same set of data might draw different, slightly different, or even widely different conclusions. There are many instances in which data that are now more and more often published as raw data, so they can be thoroughly reanalyzed by other scientists, and these other scientists draw conclusions different from the original authors because conclusions are in part subjective. Which means that conclusions are not necessarily definitive. We can believe that certain generalized conclusions are okay because they're apparently supported enough by experiments and observations, but in the future other experiments and other observations might show that there were limitations that we had not considered. So conclusions are a matter of judgment. And I'll show you a bright example of that. In the previous lesson, I've shown you a few data that, I hope you remember them, that concluded that the retinoblastoma protein is not necessary to maintain the postmitotic state in terminally differentiated cells. That is, once the postmitotic state has been initiated, it stays there whether or not the retinoblastoma protein is still expressed. Okay, that paper was published in, I think, 2006, and in, it was in 2001. No,
This is
okay.
the
This is that paper, and it was published in 2004. So there is a PRB independent mechanism that does not depend on PRB that preserves the postmitotic state in terminally differentiated cells. And a few years later, in I think it was 2007, yes, this other paper came out. The details are not relevant in this lesson, but here is one main conclusion that they drew. We show that RB is also uniquely required to maintain this arrest in myotubes. So it is a completely opposite conclusion. How do we reconcile two opposite conclusions?
One is wrong. One.
Different point
One
of
is
view.
wrong, one might be wrong, both might be wrong, both might be right, but in different conditions, in different situations, with different caveats. So, again, I won't go into the discussion, the details, which involves knowing a lot about this field. But basically, I'll draw your attention to 2 points. First, look at the previous paper. Preserve the postmitotic state in terminally differentiated skeletal muscle cells. The authors believe that probably the same mechanism holds true for other terminally differentiated cells. But they don't dare to draw that conclusion because it is not necessarily correct. So they stay in skeletal muscle cells. These other authors make a wider statement. They say, well, it's basically the same statement. It is written in a different way, but basically they're saying that the results are true in skeletal muscle cells. So again, they stick to the system they're using, and yet the 2 papers are using different systems. It is not evident in the title, it is not very evident even in the text. And yet if you read carefully, the previous paper was using primary cells. That is, cells freshly explanted from the animal. This one uses an immortalized cell line, like blasts. So, to the best of my knowledge and understanding, I believe that both papers are correct. They're both well performed, the experiments are well done, the techniques are appropriate. So I believe the data of both papers are correct, and yet the system used is different and explains the different conclusion, which means both papers inadvertently over-extrapolated, over-generalized their results. One said every skeletal muscle cell does not require B, and the other says every skeletal muscle cells does require B. And instead it's some of them do, some others don't. So this is interesting, and it is absolutely not rare. It is very often the case that papers coming to very different or even opposite conclusions come out in the literature, and sometimes, they create hard times between the authors because they're quite sensitive about their results. They don't like other people to say, no, it's not like you say, no, it's like I say. But more important, much more important than the debate among the authors is that at some point you might not know what to believe, because there is weight of evidence on both sides of the scale, and you don't know what's the truth. And sometimes, if you really want to know, because you're studying that particular field, that particular area, you have to run your own experiments and find out by yourself.
And if we try to get conclusion from the data, we can, I don't know, mediate the both articles, or we get also the same conclusion of the author.
Interesting. In this particular case, it is difficult for an outsider to determine what's the difference between the 2 papers. You have to know, as the authors of the first paper knew, that immortalized cell lines do not behave always like primary cells. The authors of the second paper also knew that, but they thought it was not significant in their case.
Okay.
It was irrelevant. But probably it wasn't. But an outsider wouldn't probably spot the difference, unless he's very smart. So this is to illustrate a real case in which we can show that the conclusions are subjective. But of course, they must always be supported by the data. But the degree to which they are supported by the data is always partial, because of the need to generalize. And generalizing is always a risk. By the way, very rarely a scientist is deemed liable because of its conclusions. You do not condemn a scientist that has drawn wrong conclusions if the reasoning was proper. That is something, a risk that everybody runs, because conclusions can always be disproven by further data, further observations. We have learned that in the very first lessons. And it is something that we should always keep in mind. So, even if it is your own data, do not stick blindly to them. If somebody else says something different, realize that probably there must be a reason. It's not that you are right and he is wrong. There might be a reason because you draw different conclusions. By the way, the first paper is mine. The second is from Alex.
The one wrong about...
yeah. So, here is another, this is, by the way, a very beautiful paper that came out in 2019 in Cell. It is so beautiful that I immediately put it into this lesson. Not only because it's beautiful, of course, but because also it teaches you lessons. In fact, recapitulates several points that we have already made. And so I'll tell you about this paper, or at least some parts of this paper, which is very complex, in order to draw these lessons from the paper itself. First of all, what is the paper about? The paper says SERT6, which is a acetylase, is responsible for more efficient DNA double-strand break repair in long-lived species. Now, the title is not so exciting, apparently. Well, if you're not interested in DNA repair, who cares about that? And what does long-lived species, what does it have to do with the previous part of the title? Well, basically what the title is saying is SERT6 is responsible for more efficient DNA double-strand break repair, which in turn is one of the reasons why species that live long do so. So this is very important. If you have a strong ability to repair double-strand breaks, then you live longer. That's a very interesting conclusion, if it is true, and we'll find out, because people have long thought that progressive accumulation of damage to the DNA, in the DNA, was one, or according to some people, even one of the most, or the explanation for aging. And yet, that had never been put to direct test, because it's very difficult to design an experiment that really tests these hypotheses. So here is a paper that really addresses the issue, and this is why it's so beautiful and so interesting. So, first of all, the authors did what I would call really a heroic effort in analyzing some, I think, 23? 19 species of rodents. Why rodents? Because the authors say they're quite different from one another in terms of lifespan. You see, the shortest lived, this is maximum lifespan, the maximum lifespan ever recorded in that species. And you have very short-lived species such as golden hamster or house mouse, 4 years. Mice usually live much less than 4 years. This is really the longest ever recorded. For a human would be 122 years. And there are long-lived species such as here, beaver, Castoro, and what is it? Oh, and the naked mole rat, which is a species often chosen for aging studies because it lives long. See,
32 years.
years. Okay, what else do you have in this table? First of all, you have a relatedness tree that shows you how close they are from one another. And the authors say, although they live very different length and their lives are very different in terms of length, they're very similar to one another. They're closer to one another in the tree of life. So they can meaningfully be compared. They also consider the body mass of the species because, according to some theories, body mass is correlated with lifespan. The heavier you are, the longer you live. But there is apparently no correlation, no strong correlation among rodents. Okay. So once they have set up this series of species, many of which were not commercially available, they had to be caught in the wilderness. Not by them directly, but they had some hunters hunt them. But nevertheless, it was not trivial to get them. So they analyzed two major systems, DNA repair systems. Non-homologous end joining, and the other system, which is not shown here, which is homologous recombination. So these are two ways to repair DNA. In non-homologous end joining, basically the cell finds two loose ends, DNA ends, and simply puts them together. So it's a kind of rough repair, sort of an emergency repair when the cell is unable to do something better. Of course, it's prone to cause errors in the DNA. Homologous recombination is much more sophisticated. You again have two loose ends, but the cell takes the other chromosome, copies it, and replaces whatever sequences are missing, if they are. So basically copies the information on the other chromosome. Now, that is far less prone to error, but does make, or at least it has the possibility to make the cell homozygous for that particular allele, because one allele is replaced by the other, by a copy of the other. All right, but never mind these details. The authors look at the ability of skin fibroblasts and lung fibroblasts in all of these 19 species to do non-homologous end joining. And in a slide I want to show, in a picture or figure I want to show you, they also look at the homologous recombination in both skin fibroblasts and lung fibroblasts. Now, these are non-trivial experiments. Doing them in a single species would be already a significant project, but they have done it in 19 different species. And of course, many times each, you see error bars here, and they find basically that there is a good correlation between efficiency of the non-homologous end joining and of the homologous recombination and maximum lifespan. Here as logarithm base 2. You see, it's pretty obvious that an increase in the ability to repair correlates with lifespan. And of course, the square R confirms. Same thing here. Why do they use two different sources for fibroblasts? Well, they basically are forced to choose fibroblasts because they are by far the easiest cells to grow in culture. And they have different species, so they cannot set up culture conditions for each one of them. That might take years if you ever succeed. So they choose the cells that are easier to isolate from the animals and culture in the laboratory. Nonetheless, they take them from two different sources because they want to be able to generalize as far as possible. So they are not sticking to just one source of fibroblasts. Good. Then they do something really impressive. There is one thing I haven't told you about. That is, in the literature, that is in the previous knowledge, there was the indication that not only the ability to repair double-strand breaks, but also another system which is called nucleotide excision repair, or NER, NER, correlated with the lifespan. But curiously, some papers reported a correlation between NER efficiency and lifespan, and others didn't. They didn't find any correlation. So again, there was a difference in, first of all, the data, but mostly in conclusion. Some papers concluded that efficiency of NER correlated with lifespan, and some other papers said that there was no correlation. So, they also looked at NER. I don't have the slide here, I should probably put it. And found that there was no correlation with lifespan. Yeah?
I'm having a thought, but couldn't it be the correlation that species that live less doesn't need a well-refined DNA repair system because they will not accumulate that much error in that lifespan?
At this point, I'll underline, we are looking at observations. Even though they require complex laboratory operations, they are basically observing. They are observing, actually getting data for the lifespan of the animals, and they're observing how efficient the DNA repair in each species is. So these are plain observations. And we have said that observations can never establish a cause and effect relationship. That is, as rightly Samuele was pointing out, we cannot say that because we have a good ability to repair DNA, we live longer. It could be the opposite. You need a good ability to repair DNA if you want to live longer. Or there might be no direct relationship between the two things, and they might be due to a third cause unknown to us. But the paper, yeah.
To observe the repair system, the inducing the double-strand breaking?
Yes. The system basically is here. It's complex, so I didn't go in it. They have this construct here, which is integrated into each cell. So it's not floating freely in the nucleus of the cell, it's integrated in the gene. Then they induce with a rare cutter enzyme called SE1, they induce a cut in the DNA, which, when repaired by the end joining, non-homologous end joining, joins these two parts of GFP. It puts them together. And so what they measure is GFP expression, which is easy to measure. GFP expression is a measure of the efficiency of the DNA repair. For non-homologous enjoining, they have a conceptually similar but different approach. But it's basically these kind of things that you do when you measure DNA repair. So, at this stage, again, based on observations, we know that lifespan, in this paper, lifespan correlates with ability to repair double-strand breaks, DNA double-strand breaks, does not correlate with nerve. Now, the authors do something really amazing. They think, they reason. Is it possible that both the non-homologous enjoining and the homologous recombination are both under the control of the same gene or the same mechanism? And based on data in the literature and based on their guess, they identify SIRT6, this assemblies, as the possible regulator. So, indeed, they measure the activity of SIRT6 in different species, and they find that it correlates very well with the efficiency of DNA repair, of double-strand break repair. And so they analyze the different protein products in the diverse species and find by a series of swaps, so they take a piece from one species and put into the protein of the other, and vice versa, in a variety of different experiments. And this is, it takes a lot of effort, a lot of time, and quite some money to do. And I find that the difference in the efficiency of SIRT6 is all in 5 amino acids that are different from one species to the next. And so they create a series of constructs in which these 5 amino acids from a long-lived species, say a beaver, are put into the SIRT6 of a short-lived species such as a mouse, and vice versa. And they find that indeed, if they do that, the strong SIRT becomes weak and the weak SIRT becomes strong. And then, and this is the majestic final experiment, they introduce these different SIRTs varieties that they created into Drosophila.
It worked.
It works. They work in Drosophila. They find it. Nobody knew about that. And it is interesting because this is now an experiment. Why did they choose Drosophila? Because Drosophilas live a couple of weeks, so you can measure the lifespan, their lifespan. And this is the results. This is a very well-controlled experiment. So, it's a bit complex. I'll try to show what it means. First of all, they have the various constructs introduced into different lines of Drosophilas. Of course, congenic lines, they're all identical. The same strain. Under the control of an inducible promoter. So the protein can be induced or not, depending on the experimental condition. If they give this drug here, the proteins are induced. If they don't, they are not induced. So, the various constructs, first of all, do not affect the lifespan of Drosophilas when they are not induced. Drosophilas all live the same amount. 50% is about not nearly 60 days. So these are long-lived Drosophilas. Okay, so we have control Drosophilas that have not been transduced with a transgene. Then we have Drosophilas transduced with mouse wild type SIRT6, mouse with beaver, with the 5 amino acids from beaver, then beaver wild type, and beaver with 5 amino acids from mouse. And when they induce the proteins, here, beaver wild type is best. The Drosophilas at 50% live about 6 days longer control is this. So they actually live longer than controls. So they have extended lifespan. And what is it? Mouse with beaver amino acids is nearly as effective as beaver, while beaver
with mouse.
with mouse amino acids is less effective, although close enough. And the worst is mouse here. All of them, though, make the Drosophila live longer than controls because all of them provide some degrees of extra SIRT6 activity. So, basically, here with an experiment in which they isolated a single variable, SIRT6, they proved, at least in this system, that SIRT6 and hence DNA repair, double-strand break repair, is responsible for extended lifespan. This is really the first proof that DNA repair controls, to some degree, the lifespan of a species. Excellent.
And
Yeah.
on the right, the control is also trained with the drug to
Yes,
induce
it
the.
is. All of
okay.
them, this
Yeah.
graph. And none of them in this other graph. So this shows that transgenesis in itself does nothing to lifespan. Here it shows that expression of what's being introduced does affect the lifespan. Good. So we come to the author's conclusion. Now, these again are subjective conclusions, although based on very serious data. They say lifespan increases according to the ability to repair double-strand break repair, double-strand breaks, DNA double-strand breaks, and that in turn this ability is promoted by the activity of SIRT6. Basically, SIRT6 is upstream of both non-homologous end joining and homologous recombination and increases the efficiency of both, the activity of both. And so there are animals that have low SIRT6 activity, and low ability to repair DNA, and live short lives. And opposite, rodents that have high SIRT6 activity live long lives. In passing, they also explain why people were obtaining different results with respect to NER. Why some people were saying that NER affects the lifespan, and others say it doesn't. The reason, most likely, they argue, based on their data and analysis of the literature, is because by mere chance. Well, first of all, I have to tell you what NER repairs. Basically, NER repairs ultraviolet damage to DNA, and nothing else. People, humans, who have NER deficiencies, have a lot of tumors on the skin, but no extra tumors anywhere in the body, suggesting that NER basically does nearly nothing other than repairing ultraviolet light. And so, according to the authors of this paper, previous authors had, by mere chance, either chosen animals that live at night, and so they're never exposed to UV light, and they are short-lived, and they conclude that there is no relation. That there is a relationship between low NER and short lives, or vice versa. Diurnal animals that are exposed to UV light, and so they need a high NER, they live longer by chance. There is no relationship between being exposed to the sun or not and lifespan. So that was the problem. That is, again, an illustration of one. The fact that by necessarily generalizing your data, you can make mistakes. You didn't realize that your data were biased. Your biased were only nocturnal or diurnal animals, and so you weren't considering an important piece of information, an important variable in your results. On the other hand, it also shows that, I'll remark it again, that observations can never establish cause and effect relationship. Here there were legitimate observations of a certain agreement between nerve activity and lifespan, and yet they meant nothing. They were all due to a common cause that was exposure to the sun. Good. But now let's consider the conclusions of the authors by going back to the title. You see, their conclusions are very general. They don't talk about rodents here. They talk about long-lived species. So it's really a rather daring generalization, which makes sense. I would agree with that. Of course, until we have further data about this. But it's nonetheless really daring because they only have data about rodents. By the way, the only experimentation has been done on Drosophilas, not even rodents. So it's really a sweeping conclusions based on data, and yet the amount of data they generated is huge. It's really hard to imagine an experiment, a study that would generate more data of this kind in a single shot. So, going back to this. By reading this paper, we have applied several lessons that we have talked about. We have considered what kind of generalization these authors do. We have considered the cause and effect relationship and the traps in which one can fall in considering cause and effect relationships when dealing only with observational data. We have considered also the difficulty and the riskiness of generalizing the data, and yet the necessity to do so. And so I think this is a very instructive paper, besides what is saying about the specifics of the topic, which is absolutely interesting. So, oops, I forgot to correct this. Data generate results. Once again, the results, we'll talk about that when we discuss the way scientific papers are written. In scientific papers, you first of all you show your results, and then you draw you show your data, sorry, and then you draw immediate conclusions called results that are the logical conclusions that you can go from the data, nothing other than, no generalization. Then later in the discussion you can generalize. Conclusions instead include this kind of generalizations, which are absolutely necessary, and yet create most of the confusion in science. If we could just stick to our data, we would have a myriad data with little meaning, because we wouldn't be able to draw laws of nature from them. But we would have very firm data, and yet we would understand very little about nature. In order to understand nature, we have to generalize from our data. So, we have said that conclusions are never unique. And then, the last thing I want to say is that from the conclusions of research work, oftentimes, most of the times, we get new questions. For example, consider this paper here. Well, the obvious question, we have already mentioned it, is it true? Does it hold in other species? is the Drosophila experiment conclusive? But more interesting, how much does the ability to repair DNA affect lifespan? In Drosophila, as we have seen, it's about 10% increase in lifespan. Is it all there? Or if we had, say, a very, very much increased ability to repair DNA, it would be much longer than that. That is there a limit to the ability of DNA repair to increase the lifespan, or is it not? If we had infinite ability to repair, the DNA would be immortal, would we be immortal? This is extremely interesting. And the other question is, if DNA repair is not the only determinant of lifespan, what other determinants are there? What is relevant to lifespan? What other mechanisms affect lifespan, determining whether a species lives longer, whether a single individual lives longer than another of the same species, and so on, and so on, and so on. So this paper is also beautiful because it generates a lot of questions. But that's typical. Answers in science generate more questions than answers in science. So, this lesson was about interpretation of results, and here are the highlights. The detailed design of experiments follow the identification of the experimental approach. Oh, this is about the whole cascade of levels concerning the scientific study, scientific project. And then now, concerning this particular lesson, raw data must be translated into results, logical conclusions from the data. In the case of the microarrays, the logical conclusion was the arrays are not sensitive enough for our purposes. From results, one draws conclusions. But remember, conclusions are subjective. They must be drawn based on generalizations, and generalizations are risky. A good experiment always tries to tell us something, even if we don't understand it, or even if we don't want to hear it. And so, a valid result should not be denied, unless you should kill the generator of the result.